The next group of antibiotics we're going to take a look at damage the plasma membrane. So we've got the polymyxins, which include polymyxin B and colistin, and there's also daptomycin, but you don't need to know that one. So the way that these antibiotics work is they will poke holes in the bacterial plasma membrane, and then that will end up killing the bacterial cell. Polymyxin B ends up poking holes in the bacterial cell membrane, like we said, and it's actually just mainly used topically, and it's part of Neosporin. So if you look on the Neosporin that you have at home, you'll see polymyxin B is one of the antibiotics in Neosporin. And the other antibiotics, we have neomycin, which that's an aminoglycoside. Remember, aminoglycosides will bind to the 70S ribosomes and inhibit protein synthesis. And then we also have the third antibiotic in Neosporin is bacitracin, and that one inhibits cell wall synthesis. So Neosporin with the 3 antibiotics in there gets a lot of things. The other antibiotic that will poke holes and damage the plasma membrane of bacterial cells and kill them is colistin. Colistin is a very old antibiotic from the 1940s, and it's one of our big gun antibiotics. It's an antibiotic of last resort. So we don't like to use it unless nothing else is working, and maybe colistin will still work, although we are starting to get resistance to colistin as well. So the reason that we stopped using colistin way back in the 1940s is because it had a lot of side effects. So it can damage the kidneys and cause kidney failure, and it can also cause nerve damage as well. So it can have some pretty toxic side effects. All right, our next group of antibiotics will inhibit nucleic acid synthesis. And we've got ones that will block DNA replication. And these are the quinolones. What enzyme that they inhibit is topoisomerase. And if you remember, during DNA replication, topoisomerase is the enzyme that runs in front of helicase, and it undoes all the knotting and the twisting in the DNA. And so if you block topoisomerase from working, then the bacterial cell can't replicate its chromosome, and then it can't divide, and that's a problem. And then the other nucleic acid synthesis inhibitor is the antibiotic called rifampin, and it's going to block transcription. And remember, in transcription, you're making messenger RNA. So they're not able to make the messenger RNA, and if you can't make messenger RNA, then you can't make the protein. So the enzyme that rifampin will inhibit is RNA polymerase, because that's the enzyme that does transcription. And rifampin is used to treat mycobacteria. So that would be used to treat, for example, TB and leprosy. And rifampin is usually never used by itself. It's usually used in combination with other antibiotics. And it can also be used to treat some other bacterial infections besides TB and leprosy. So sometimes you might see it used to treat some of the resistant bacteria along with some other antibiotics as well. An interesting side effect with rifampin, if you look at the capsules here, you can see that they're kind of a red color. And the side effect that we see with rifampin to warn patients about is that it will turn any body fluid orange. So their urine turns bright orange, it colors the stool orange, their tears, saliva, and even their sweat will turn orange. That would be pretty alarming if you didn't know it did that. And also, I don't know, it'd be a little embarrassing to go to the gym and have orange sweat. So, okay, so here's an example of the quinolones that inhibit DNA synthesis. So they block the enzyme topoisomerase used in DNA replication that undoes all the knotting and the twisting. Most of the quinolones that are used today are fluoroquinolones, so they've just attached a fluorine atom to them. Probably the most famous quinolone or fluoroquinolone that you might be familiar with is ciprofloxacin, or sometimes called Cipro for short. So you may have seen that used in patients before. Okay, and then just looking at the chemical structure, and you can see the fluorine atom that's been added to these quinolones to make them fluoroquinolones. Our next group of antibiotics are called metabolic antagonists or antimetabolites. There's two of them in this group. We have sulfonamides or sulfa antibiotics and trimethoprim. So the way that these work is they will block metabolic pathways by competitively inhibiting enzymes. And remember that competitive inhibitors, how they work is they look very similar to the substrate that normally will bind to the active site on the enzyme. And so they will end up competing for the active site on the enzyme, and if the competitive inhibitor binds there first, that prevents the substrate from binding to the enzyme, and that blocks the enzyme from catalyzing the chemical reaction. So that's how a competitive inhibitor works. All right, so the sulfonamides or sulfa drugs, they look very similar to the substrate called para-aminobenzoic acid, or PABA for short. And the sulfonamides will compete with PABA for the active site on an enzyme that's involved in folic acid synthesis. And folic acid is actually really important in bacteria because it's going to be needed to synthesize purines and pyrimidines. And if you remember, purines and pyrimidines are part of nucleotides. So they're the A's, T's, C's, G's, and U's that make up part of the nucleotides. So remember, we need the nucleotides in order to make the DNA and the RNA. And the next question, do sulfonamides or sulfa drugs affect our cells? And the answer is no. And the reason is because we don't make folic acid. We actually get folic acid from our diet. So we don't even have this enzyme. All right, so here's just a picture of how the sulfonamides work. So here's the bacterial enzyme in green with the active site. Here's the normal substrate, PABA, in blue. And this enzyme will convert the PABA into the next thing, and then eventually it will turn into folic acid, which will then be used to make the purines and pyrimidines, part of the nucleotides, which the bacteria need to make its nucleic acids, the DNA and the RNA. So if you're giving your patient a sulfonamide antibiotic, it looks very similar to the PABA, that normal substrate in blue. And so if you give enough of the sulfonamide antibiotic, it will outcompete the normal substrate, the PABA, and the sulfonamide antibiotic will bind to the enzyme's active site and get there first, And then the normal substrate, the PABA, can't bind to the enzyme, and so that stops the chemical reaction from occurring, and the bacteria can't make the folic acid, which means it can't make its DNA and RNA. So then it's in trouble. Okay, and then the other one that's also working very similar to sulfonamides would be trimethoprim. It's also going to work the same way. It's a competitive inhibitor, and it also works in the same pathway. So it's just going to compete for a different enzyme in the pathway and also inhibit folic acid synthesis. And what we do is we actually will combine the sulfonamide and trimethoprim, put them in the same pill, and give the patient both of these together. So you can see this bottle here has both a sulfonamide and trimethoprim together in the same pill. And so by taking those together, we're actually going to be blocking 2 steps in the folic acid pathway. And that way we doubly make sure that the bacteria can't make their folic acid, and then they can't make the purines and pyrimidines or their nucleotides, and they can't make their DNA and RNA, and then they're going to be in trouble. By using both of these together, they act synergistically, and what that means, syn means together and erg means to work. So they actually, by having them be given together, you actually get a much bigger effect if you have them given together than if you just gave them just one of them individually. So it's better to give them both together than just one of them. So that's usually what we do. The brand name of these drugs here that are combining the sulfonamide and the trimethoprim, you might have heard of Bactrim or Septra. Those are some of the brand names of these. Okay, and then this just shows the metabolic pathway for taking the PABA substrate that we start with. And then here's the sulfonamides blocking this first enzyme in the pathway. And then here's trimethoprim, and it's going to block the second enzyme in the pathway. And both of these are competitive inhibitors, competing with the substrate to block those enzymes. And so if we block both steps, we definitely are going to be blocking the synthesis of the folic acid, and then they won't be able to make their purines and pyrimidines and be able to make the nucleotides and the nucleic acids. Okay, and then just some other antimicrobial drugs. I know we've been focusing a lot on antibiotics, but we do have some other microorganisms that we need to treat in patients. So we do have some antifungal drugs. So antifungal drugs have a low therapeutic index. And a lot of times we have some problems with toxicity and side effects. And remember that the therapeutic index, how we get that is we're going to divide the toxic dose divided by the therapeutic dose. And remember we said that we wanted a high therapeutic index, we wanted a high number, and this was just a measure of drug safety. And if you had a low number, if you had a low therapeutic index, that just means that your toxic dose and your therapeutic dose are pretty close together. So like we gave that example where maybe your toxic dose was 2 mg per kg and your therapeutic dose was 1 mg per kg, and that gave you a therapeutic index of 2, which was a pretty low number. And remember we gave the example where maybe the therapeutic dose was taking 1 pill, but the toxic dose where we start to see side effects was 2 pills. So, and that, could happen where a patient could forget they took their pill already and then they took another one. So the reason that we've got a low therapeutic index with these antifungal drugs, why the toxic dose is pretty close to the therapeutic dose, is because fungi are eukaryotic cells like our cells. So these antifungal drugs, in targeting and trying to kill the fungi, a lot of times they're going after things that our cells have as well. So they end up, sometimes damaging our cells as well, and then we get side effects. Okay. It's much easier to treat superficial mycoses, and a mycosis is a fungal infection. So superficial ones would be ones that are, in the skin or hair, nails, than ones that are systemic, which means they're internal, okay? Because the superficial ones you can just treat topically, like maybe with a cream here, whereas the ones that are systemic internally, maybe you've got a fungal lung infection, yeah, you've got to take drugs internally, and then you're going to run into a lot more side effects. The antifungal drugs work in a lot of different ways. You do not have to memorize any of these. Okay, you can look at them, but I'm not going to ask you that. Okay, so just treating mycoses or fungal infections. Just some examples of superficial mycoses. We have ringworm, which is pictured on this child's forehead. It's kind of a bad name. It's not caused by a worm, but it's a fungus. And so that could just be treated with a topical cream or ointment, antifungal. And then candida, that's a yeast. So like maybe a thrush in the mouth, that's a yeast infection in the mouth, or maybe there's a vaginal yeast infection, or maybe there's a yeast infection like in a skin fold or something. That could be candida as well. So those that are just, on the skin or maybe in the hair, those can usually just be treated with some topical drugs, and then you don't have to worry about those side effects because it's not gonna get internally, and so you don't have to worry about the side effects. But the systemic mycoses, where the fungal infection gets inside of you, those are going to be more difficult to treat because you've got to give these drugs internally to treat the fungal infection. An example of that would be something called valley fever, or also called coccidioidomycosis. This is a fungus that lives in the soil, and it's found in the deserts like around Phoenix, Central Valley of California, and you breathe this in like maybe in a dust storm or something, and then it can get in the lungs, and if you're immunocompromised, then it can cause pneumonia. So you've got to take some of these drugs internally, and they can have a lot of side effects. So that's not good to have to take those. Okay, you can just look at this, but there's just a picture from your book just showing some of the mechanisms of how these antifungal drugs work and just some of their targets on the fungus that they attack. Okay, antiprotozoan drugs. So do you think antiprotozoan drugs will have a high or low selective toxicity? And remember, selective toxicity means that you want it to kill just the microorganism and not hurt the patient. So do you think they're going to be pretty good at just killing the microorganism and not causing side effects, or not? So if they have a high selective toxicity, that means they're pretty good at just killing the microorganism and not causing side effects. And antiprotozoan drugs have a low selective toxicity. Okay, so that means they're not so good at just killing the microorganism and not hurting our patient and causing side effects. So the reason that is because protozoans are eukaryotes like us. And so these drugs, in targeting and trying to kill the protozoa, they're going to go after things that the protozoa have that maybe our cells have as well. And so they could end up damaging our cells too. Okay, there's a lot of different protozoans that cause infections in people. I've just got a couple of examples here. So like malaria, and I've got a couple different drugs that can be used to, there's a lot of different drugs used to treat malaria, and unfortunately the Plasmodium protozoan that causes malaria is becoming very resistant to a lot of our drugs, so it's getting very difficult to treat. And then Giardia, one of the treatments for that is metronidazole. So one of the brand names is Flagyl, so if you work in clinics, you might have heard of that. And metronidazole is interesting because not only does it get protozoans like Giardia, but it also can kill bacteria too, which is interesting. Okay, and last thing would be antiviral drugs. It's pretty hard to develop antiviral drugs that are selectively toxic and that only get the viruses and not hurt our cells as well. And the reason that is I know we haven't talked about viruses yet, but viruses, in order to reproduce, they take over our cells' machinery to produce more virus. And so if you're trying to target the virus, you may end up targeting our cell because they're using our cell's machinery in order to reproduce. Okay, so you have to be careful what you pick when you go after the viruses, because you don't want to go after something in our cell. Okay, so the antiviral drugs that we have, they want to look for something that is just specific to the virus and it's not something that our cell is using. Okay, and so we'll talk about some of those things when we get to viruses. And the other thing that you'll find is that when we do have an antiviral drug, they're usually pretty specific as far as which virus they're going to get. We don't have broad spectrum antiviral drugs. The reason that is because, as we'll learn later, there are 7 different groups of viruses, and they all have different life cycles or ways that they reproduce, and so there's no way that you're going to find an antiviral drug that will, get a lot of different groups. So, because they're all very different. Okay. All right, so this is just showing you a viral life cycle, and this is actually just for a bacteriophage, a virus that infects bacteria. But all I just want you to get out of this is just we need to look at these viral life cycles, how they're reproducing inside the cell, and look for something that is just specific to the virus, and it's something that our cell is not using, and we want to target that. Okay, so that's what these antiviral drugs are going to go after. Okay, so maybe there's some enzyme that's specific to the virus that our cell's not using. We can target that, for example. Okay, so these are just some examples of antiviral drugs. You don't need to memorize any of these that are not highlighted in red, but you can just look at them. But notice that they're pretty specific as far as which viruses they're going to get. So they're pretty narrow spectrum. The other one that's in red, I do want you to know because we're going to come back and talk about this one later. So interferons, these are interesting because these are actually produced by our body cells. And they're produced by when our cells get infected by a virus. So let's say we have our body cell here. And if it gets infected with a virus here, here's our happy virus. And this cell says, oh my gosh, I'm a goner, I'm infected. Well, this infected body cell secretes these chemical messengers called interferons that will warn the neighboring cells to get ready that a virus might be coming their way. And so these neighboring cells will do things to get ready for a possible viral attack. Okay, and so do things to prevent the virus from reproducing in them. So we'll talk more about them later, but just remember that term. Let's take a look at Tamiflu, an antiviral drug you probably have heard of. So this one will target the influenza virus. And what it does is it inhibits an enzyme on the influenza virus called neuraminidase. This enzyme is located on the surface of the flu virus. It's this green, what we call a spike protein here. And what it's used for is after the flu virus has reproduced inside your cell and made a bunch of new flu virus, the Tamiflu will prevent the neuraminidase enzyme from releasing this newly made virus from your cell. Okay, so then it just stays attached there and it can't be released and go infect another one of your body cells. So it just helps you recover from the flu a little bit faster, maybe like by about a day, and that's about it. Unfortunately, we are seeing resistance to Tamiflu. So we see resistance to everything. So not only do we have resistance to the antibiotics with the bacteria, but we've got resistance with the viruses, to the antivirals, resistance with the fungi, to the antifungal drugs, and we also have resistance with the protozoans to the antiprotozoan drugs as well. So everything develops resistance. And so we do have resistance with Tamiflu as well. So we're trying to reserve it and just use it for the people that really need it, like those that are elderly or really young or maybe immunocompromised.